Welcome to Biol102. This is Dr. Siddiqi, and today I am going to talk about the cell structure and function. Today I'm going to talk about the cell structure and function. And here, the before I start with the cell structure and function, I want to tell you that there are several branches of plant sciences. This is what you have studied in diversification of botany. And the branch that deals with the structure of cell and function of cell is called cell biology or cytology. So let us start with cell structure and function. And you're looking at these slides. You already have set of these slides, and I will use these slides to explain the cell structure and function. Those of you with odd roll numbers have already learned this in the lecture room, and those with even number will learn it with the help of these slides. If there are any questions after you hear this recording, you are welcome to ask me these questions. So let us start with cell structure and function. And the first slide here shows the ultrastructure of cell, and we are going to talk about the various parts of the cell. But before we do that, let me start with the history of cell biology. Cell history. And here, when we talk about the cell history, there are several names that you are supposed to learn. The first one is very important, and this is called Robert Hooke. Robert Hooke in 1665 discovered cells. So he's called father of cell biology or cytology. He's the one who discovered cell. What he did was he took a piece of cork and then cut a very thin section of this piece of cork and looked at it under the microscope, and he found that there are chamber-like structures under his microscope, and he called them cells. So this is how cells were discovered. Then there are several other names: Malpighi, Grew, and Leeuwenhoek. They also reported cells in various plants. The next name is Lamarck. Lamarck in 1809. He said that cells only come, there are no life without cells. So there was this debate of biogenesis and abiogenesis, that living things come from other living things or living things come from non-living things. So Lamarck, 1809, for the first here said that biogenesis is true. Pasteur also worked on this hypothesis of biogenesis, the theory of biogenesis. Then the next name is Dutronchet, and Dutronchet said that all animals and plants are made up of cells. Robert Brown is very important name, and he discovered nucleus and nucleolus. He discovered nucleus and nucleolus. And then there are these two names, Schleiden and Schwann. They are the one who formulated cell theory. In the next slide, you will learn what this cell theory says. Then there was this Virchow. He added to the cell theory, and he talked about the origin of cell. Then there is this Buchner, and he talked about fermentation. In the next slide, you see this Hooke's microscope. This is a very simple microscope. The microscope that you will see in the lab is modern microscope, compound microscope. This one is a simple microscope which was used by Hooke when he looked at these cork cells. So it has an ocular and it has an objective. Okay, so this is what cell theory says. Schwann and Schleiden and Schwann came up with cell theory, which says that all living things are made up of cells, and Virchow added to this cell theory, and he said that all cells come from pre-existing cell. He was a French cytologist, and in his French he said cellula e cellula, which cell means that all cells come from pre-existing cell. In the next slide again, you see the various parts of the cell theory. All living things are made of cells. Cell is the smallest unit of structure and function. All cells arise from pre-existing cell. So this is these are the important points of cell theory which were formulated by Schleiden and Schwann. Then these various microscopes, and those with odd roll numbers already had this this inquiry activity on microscope. You have seen the light microscope, you have used it, you know how to use it, and you also seen the dissecting microscope. The other group will have a look at this light microscope. Electron microscope, we don't have it, but you use beam of electron which pass through a tube. There is a phase contrast microscope, a transmission electron microscope, and the scanning electron microscope. These are the various types of microscope, but you will be using the first type, the light microscope. Here on the left hand side is a compound light microscope, the type that you will use in the lab. On the right hand side is stereo microscope or dissecting microscope. You will also have a look at these two types of microscope. This is a picture of electron microscope in which you use a beam of light. Okay, then we talk about the cell size and cell shape. They are microscopic. Almost all the cells are microscopic. And here, to give you an idea about the size of cells, it says that 30 plant cell will fit across the head of a pin. And there are some cells which are big in size. Fiber cells are 20 centimeter. There is this alga acetabularia is 2 to 5, but most of them are small, and you can see them only with the help of microscope. Now the question is, why cells are so small? Why cells are so small? And the answer to this is that this is because of large surface area to volume ratio. And with these small cells, you can have more efficient communication. There is variety in shape of these cells. They are spherical, rectangular, oval, box-like, and tabular. So these are the different types of cells, cell size and cell shape. This gives you an idea about cell size, and here you have to learn two terms. One is micrometer or micron. Earlier it was called micron, now it is called micrometer. Micrometer is 1/1000th of a millimeter or 0.001 millimeter. So when you see something under the microscope, you measure it in micron, 1/1000th of a millimeter. Then there is a term which is called angstrom or nanometer. Nanometer is 1/1000th of a micrometer. And here it gives you the size of these cells. You can see with the unaided eye, you can see something as small as 0.1 millimeter. If it is smaller than that, you can go to light microscope, which can see up to 200 nanometer. And if it is smaller than that, then you have to go to electron microscope, which can go down to 0.5 nanometer. This picture shows a plant cell under light microscope, and this is what you will see. Some of you have already seen it. The other group will see it on next week when you come for the lab activity. This is again a cell under electron microscope. This is how you see it. It's much bigger, much more magnification. And here is a plant cell under electron microscope. We will describe the various parts one by one. This is an animal cell. I will tell you later on the differences between plant cell and animal cell also. There are some differences, there are some similarities, and we are going to talk about this plant cell and animal cell. There are these two main types of cell. These two terms are very important, prokaryotic and eukaryotic. Prokaryotic are those cells which do not have organized nucleus. They do not have the organized nucleus. They are called prokaryotic. And they do not have organelles. The eukaryotic cells have an organized nucleus. What we mean by organized nucleus, it has a nuclear membrane, nucleolus, but in the case of prokaryotic, there is no nuclear membrane, there is no nucleolus. So unorganized in the case of prokaryotic, organized in the case of eukaryotic. And prokaryotic cells have no organelles, eukaryotic cells have organelles. Okay, this next slide tell you why cells are so small. So here you can see surface to volume ratio, and here they have more surface area. And the second thing is they can have efficient transportation with the help of the small cells. So you have to remember, cells have large surface area to volume ratio, and then you can have much more efficient transportation with the help of smaller cells. Next, we go to cell wall. This is the outermost layer of the cell, and this is made up of cellulose, hemicellulose, lignin, pectin, and glycoprotein. So you should have some idea about the major types of compounds which are found in the cell. Carbohydrate is one of them. And these are all carbohydrates: cellulose, hemicellulose, lignin, pectin, glycoprotein. This is the chemical composition of cell wall. Cell wall has these cytoplasmic strands which run through it, and this is called middle lamella. There is primary wall, secondary wall. So secondary wall is 3-layered, as I will explain to you when you come in the class. Plasmodesmata are the cytoplasmic strands, and they are used for communication. They are used for communication. Here in this slide, you can see the ultrastructure or electron microscopic structure of cell wall, and they are made up of the cellulose fibers which have a crisscross arrangement. Crisscross arrangement means these fibers will run in this direction, and the other layer will run in the opposite direction. This is called crisscross arrangement. Here you can see the cell wall with the middle lamella and the primary wall and the 3-layered secondary wall. Okay, this is the end of this recording. So you have learned up to the cell wall, and next we are going to talk about the other parts of the cell. So this is first set of slides that I have discussed with you. In the next recording, I will discuss the other sets of slides, the second sets of slides on cell structure and function. Thank you for listening.